Okay, so thank you, David, for agreeing to give this Zoom colloquium. It's a real pleasure and honor to introduce David, who some of you may know was my PhD advisor. So, I learned a great deal from him, and that's influenced my entire research career, even though I'm not quite working on the same fields. So thank you, David. So David got his PhD at Cornell and became a junior fellow at the Harvard Society of Fellows in 1975 and has been at Harvard essentially since then. He's currently the Arthur K. Solomon Professor of Biophysics and Professor of Physics and Applied Physics. So among his achievements, his earlier work was more in statistical physics as applied to what now called soft matter. He won the Oliver Buckley Prize from the American Physical Society for that work. I'll read the citation. For seminal contributions to the theory of condensed matter systems, including the prediction and elucidation of the properties of new partially ordered phases of complex materials. Please mute your mic. Maybe I should read that again. For seminal contributions to the theory of condensed matter systems, including the prediction and elucidation of the properties of new partially ordered phases of complex materials. And this includes something called the stacked hexatic phase of liquid crystals, which was predicted by David and subsequently observed in experiments. He's also won the Bardeen Prize for research in superconductivity. That was, I think, work on vortex liquids and various superconductors. More recently, David's interests have been in the biological directions, and we're going to hear much more about that today. So welcome, David.
Thank you, Subir. And while I'm attempting to share the screen here, I'd like to personally thank you and Stephanie and all the wonderful staff at the Harvard Physics Department for keeping us going during these difficult times. I myself have been a chair, but I certainly never recall signing up for the kinds of unanticipated challenges that Subir and his staff have been overcoming wonderfully. And I'd also just like to say that that stacked hexatic phase was the direct result of my collaboration when Bert Halpern was my mentor. So it's great to have him in the audience as well. So I'm going to try to tell you a little bit about some of the work that I've been doing with some of my friends and collaborators, some of whom are in the audience. I'll say their names a little bit later. And the motivation for this work is that in all probability, life, possibly even relatives of the coronavirus, probably involved first in a liquid environment. And in fact, if we, I think you can all see this fake laser pointer, 2 to 3 billion years ago, like today, water covered most of the Earth, and presumably in some tide pool, the other life got started. Once cellular life got started, some of these microorganisms, which might have been bacteria, for example, went into deeper waters. And we have evidence in the fossil record for oxygen-producing cyanobacteria. They actually invented photosynthesis that go back maybe 2 billion years. And once they invented photosynthesis, sort of like the Anthropocene era that we're living in, where humans are changing the environment, they too changed the environment by producing copious amounts of oxygen as a waste product, killing off a lot of the life around them, but also setting the stage for organisms that could exploit oxygen, ultimately leading to multicellular organisms like ourselves. And the spatial growth and evolutionary competition of organisms like this cyanobacteria Senecacoccus took place in liquids. at both high and low Reynolds numbers. I'll remind you what the Reynolds number is a bit later. And it's known that these photosynthetic organisms, especially in the somewhat deeper ocean waters, control their height to resist downwelling currents and stay close to the ocean or lake surface where they can have access to sunlight. Here's a picture of a cyanobacterial bloom in a lake. I think it's maybe a satellite photograph in Guatemala. And you can see these streaks, these green streaks. It sort of looks like the early stages of cream being mixed in your coffee. But interestingly, it rarely gets mixed to uniformity. It typically has these striated patterns. And of course, unlike cream in your coffee, these organisms are reproducing. They typically divide every 12 to 24 hours. Here's a plankton bloom off the southern coast of Iceland. Here's a 110 kilometer length scale, so this is quite a large length scale. The dimensionless number that controls the solutions of the Navier-Stokes equations is a size, like this 110 kilometers, times a typical velocity, maybe it's a wind-wave interaction that's producing a velocity, divided by the viscosity of water, which is about 10 to the minus 2 in CGS units. And if you put all those numbers together, you'll get a Reynolds number, which is the fundamental nonlinear coupling constant in fluid mechanics of something like 10 to the 8th or 10 to the 9th. It's enormous, and it produces what we call a Kolmogorov cascade of eddies, from large eddies to smaller eddies, all sorts of intermediate time scales and length scales as well. The large eddy turnover time, typical time scale for oceanic weather, is a couple months, so that's like 50 days. Small eddy turnover times go down to 5 minutes, and then you have time scales, everything in between in this theory of turbulent energy transfer from large scales to small scales. And the plankton doubling time, similar for the Seneca caucus, is right in the middle of this Kolmogorov cascade. So some of the eddies look very quick, and they can sort of average over them in a lifetime, but some of them are very long-lived, and then the organisms have time to divide and change their behavior in response to the fluid flow. So that's the sort of what got us interested in this problem. We just tell you a little bit about a very simple problem called the turbulent advection and diffusion of a passive scalar. Here's a plume-like emission for pipe turbulence. Looks a little bit like cigarette smoke, perhaps. Here on a much bigger length scale is a volcanic emission in Iceland. This is a 125 kilometer swath of the ash cloud from this Icelandic volcano with this unpronounceable name. If anybody can pronounce it, perhaps you could tell me the correct pronunciation in the question period. It's spreading out in an interesting way, subject to very high Reynolds number turbulent diffusion, if you like. And if you just have a simple system like this, an ash cloud or maybe smoke from a cigarette, and here's the concentration of the particles involved, C of X, then it's known that there's advection in the velocity field U, and then there's diffusion. It's typically incompressible flow, so the divergence of this velocity field is 0. There's something like a Reynolds number called the Peclet number, it takes a few large eddy turnover times, and eventually these things are sort of mixed together. At least that's what happens to cream in our coffee. And the standard way of arguing that is to just take this nonlinear, this bilinear equation actually, in the concentration and some external velocity field, could be the atmospheric turbulence in the Earth's atmosphere, multiplied by 2 times c, and do some manipulations. You discover that c squared, the square of this concentration, averaged over all positions, say in a teacup, we'll take that as an example, is given by the gradients of c squared times the diffusion constant. And so this says that this c squared has no alternative but to go down. It goes down rapidly, and it gets mixed to uniformity very quickly, as I said. Microorganisms living in a watery environment behave very differently. For one thing, they reproduce. And so if we wanted to try to understand their dynamics, their concentration could be affected by some background, possibly turbulent velocity field. There'll be diffusion. But I've now added a kind of logistic equation piece on the right-hand side where mu is a growth rate. So if this were the coronavirus in Massachusetts, mu would correspond to a doubling time of about 10 to 12 days. We hope that will become much longer. And then there's a saturating term in many of these microorganism equations, which is often modeled as 1 minus C, so that the organisms grow up, they reach a saturated carrying capacity in a nutrient-rich environment. So this is an important change from the way cream mixes in your coffee. But another important change is that the effective velocity field acting on organisms near the ocean surface is in fact compressible. It's a 2D effective compressibility, and the reason for this particular change is illustrated down here. This is a cartoon of what goes on, say, in the first 50 meters of the ocean, where organisms can, in fact, get access to the photons for photosynthesis, and they control their buoyancy to try to stay near the surface. And as a result, although there may well be downwellings and the kind of three-dimensional stratified turbulence in the ocean, they tend to resist the downwellings, and they get accumulated at these, between these pairs of rollers in this simple cartoon model, and they tend to be swept away from upwellings and accumulate at downwellings. And that effectively means that there's a 2D velocity field when averaged over the 50-meter thickness of the ocean, which has a non-zero 2D divergence. And this combination of these two effects and the red boxes really changes everything. And in particular, here's the simulation with my collaborators, Prasad Perlikar, Federico Toschi, and Roberto Benzi, where this is the heat map here is just the concentration of some particular microorganism, desperately trying to grow up, but also being scrunched together by basically compressible 2D turbulence at Reynolds number of about 100,000. So the way this is done is you take a cube of 3D incompressible fluid, and you allow the organisms to live on a slice of this fluid, say the midsection of a cube with periodic boundary conditions. They resist going out of their preferred depth, and so you look at the projected velocity field, and it has a non-zero 2D divergence. And unlike cream in our coffee or whatever, this actually never settles down. There's a statistically steady state, but it never gets mixed to uniformity because the compressive flows are always scrunching the organisms down to high densities. In fact, densities greater than the carrying capacity, which has been rescaled to 1 in this model. And then they go crashing down when they get bigger than the local carrying capacity. So you can have a lot of fun with high Reynolds number fluid mechanics and a little bit of biology by forming these dimensionless numbers like the Reynolds number, there's a Schmidt number, but there's an important new number here that comes from the biology, which is the doubling time of the organisms, which might be 12 hours or 24 hours for these marine organisms, divided by, say, the small a turnover time. So that's a dimensionless ratio of times, and the interesting physics happens when this doubling time is large compared to the eddy turnover time, so that small compared to the eddy turnover time, so the organisms can do interesting things. I want to put one more layer of complexity before turning to the main subject of my talk, and then I'll ask for questions, which is what happens in a Darwinian sense when organisms compete under these conditions, which surely happens in the ocean. Very similar organisms perhaps, but with slight genetic modifications, might be undergoing natural selection or just fluctuating in their competition. And that's shown here, where I'm going to show you a simulation momentarily where we have organisms, say red one and a green one, and their world lines, or if you like, their Feynman diagrams, look like this. Space is horizontal, time is vertical. Here's a cell division where one red organism just divides and becomes two red organisms, similarly for green. Black could be either red or green, and these world lines indicate that death will happen when the organisms get too heavily concentrated in one point in space for the nutrients to actually support them. This is a simple example where we have neutral organisms. They divide at the same rate. Don't have to do that. Abby Plummer, who's my student, has been studying what happens when that's not true. This is sometimes called survival of the luckiest. Okay, and so I'm going to get out of this laser pointer mode and see if I can play this movie. There we go. So we started off with lots of little red and green organisms. This is an agent-based simulation. And so we're not solving a partial differential equation for a concentration. We're actually tracking, allowing these cell divisions and cell deaths in, however, a turbulent flow at Reynolds number of about 10 to the fifth. And we're letting our competitive organisms fight it out. And you might imagine that under these conditions, one or the other of these guys is going to win eventually, just because of stochastic number fluctuations. If I stop the simulation at this stage, most audiences think that the green is going to win. At least that's been my experience. But if I go far forward in time, you can see that red wins. And since we don't have mutations between red and green, it's all over for the green at this point. But basically, very little is known about what is the power of natural selection, if one has a selective advantage under these conditions. You have to know something about fluid mechanics at high Reynolds numbers and also something about population genetics. And for a long time, I've dreamed of finding simple, repeatable laboratory experiments, not simulations, which would allow us to explore these questions in a simple way. To explore, for example, spatial population genetics in some fashion. High Reynolds numbers like are present in the ocean might be a little hard to achieve in the laboratory, but low Reynolds numbers are quite biologically relevant. But what we want to do in these hypothetical experiments, they're no longer hypothetical, I'm glad to say, is to impose flows on organisms with reproduction times, it's 1 over this doubling time, if you like, that are much smaller than an eddy turnover time, so that they have a chance to adapt to the flow. And for a while, we thought, well, maybe we'll have pumps and syringes and make our little flow in the laboratory. It might be low Reynolds number, but set up these conditions and see what happens in this competition. So that's the introduction. Any questions on what I've said so far? Please don't hesitate to interrupt.
I have one question.
Please.
So basically, in the first examples, you were showing the phytoplankton bloom or bacteria, but
Yes.
then you're not using any sort of motility. You're just saying that those are transported. Yes. But
that's right.
Yeah. So Roman Stoker showed that you have also these gyrotaxis or rheotaxis that will last also. Yeah. Yeah,
that's a very good question. And some of these organisms have motility, and as Roman et al. Studied, and some of them. But in any case, there's one simple way of modeling that is to take this diffusion constant for the organisms and to simply enhance it. If one does a la Howard Berg, for example, with bacteria, a run and tumble random walk that can be approximated by a diffusion constant that's maybe 1,000 times bigger than what you might have guessed from the Stokes-Einstein relation. So you can certainly put it in, but you can also just approximate it by putting in a larger diffusion constant. And we're looking at very big scales. And I would say, in contrast to those beautiful experiments by Roman Stocker, we're looking at much longer time scales. To the best of my knowledge, he doesn't look at what happens when you have cell divisions, or at least has not done that extensively. So that would be my answer. Any other questions?
Yeah. In your, where you have, what do you call it, evolution, you know, you have your red and your green, that's a very simple model of evolution, right? I mean, undeniably. And I wonder what happens when you get more complex, when, you know, when you have further mutations, so that you don't end up actually with the dominant, because...
Absolutely, yeah. And that's a beautiful and very interesting question. I just wanted to motivate these experiments. I just wanted to show you as the simplest case. We have begun to look at what happens when you have mutations. We've begun to look at what happens when you have a selective advantage. And it's a question of timescales. And again, on very long timescales, you wouldn't just, of course, as you know, go from red to green. There would be a billion different genotypes that would be competing, and that's something we would be very happy to put in. But I'm going to take some baby steps now, and we'll hope to worry about mutations at some later stage. Okay, so let me tell you about some experiments on hard agar plates that will motivate what I'm going to tell you about. This is an experiment that actually grew out of a rotation that I did as a ersatz graduate student, even though I was probably in my mid-50s at the time, in Sharad Ramanathan's lab, where actually Kevin Foster and Zhao Xavier put down a mixture, a 50-50 mixture of this blue genotype and this yellow genotype, and watch them expand in this radial fashion. These were as genetically neutral as they could make them. And as you come out of this inoculant area, we sometimes call that the homeland, as you come out of there, in fact, you get this genetic demixing into the two genotypes. And that's just pure genetic drift, pure number fluctuations. And here's some of the experiments I actually did with E. coli, spreading again on a hard agar dish. There's no fancy fluid mechanics here yet. And these E. coli, the red and green strains, genetically demix as well. There's this halo during the early stage inflation. It's an inflationary 2D universe here that we're looking at. We can do this experiment over and over again, and we can, if you like, replay the tape of life in this simple Petri dish context on hard agar. And then with a certain radius, R0, you can come out and count the number of sectors, maybe infer backwards, see how good your inference model is of genetic drift to determine the radius of this homeland and so forth. That's background. And with that very lengthy introduction, I want to now talk about the main subject of this presentation, which is very recent work on the growth and form of microorganisms on liquid substrates. And you'll see that they have a very complex colony morphology, which strongly influences, we believe, their evolutionary dynamics. Here's some of the pictures that I hope to explain. And here are the people who actually did the work. On the right is my longtime collaborator, Andrew Murray. And then I think in the audience are both Brian Weinstein and Severine Addis, very talented grad student and postdoc who really did the hard work and made this thing actually happen. And I think it probably took of order 3 years to figure out everything that was going on and to do all the tests and eliminate various possibilities and so forth. So I'm deeply grateful, especially to those two. And also thank you to the Harvard MRSEC, which stands for Materials Research Science and Engineering Center. And believe it or not, they actually supported some of this work, even though it's a priori not traditional material science. So I'm quite grateful. And you might imagine that a card-carrying theorist like myself, it's not so easy to find funding for this kind of exercise. But I think something rather nice came out. Okay, so it turns out that we didn't need pumps and syringes. Microorganisms grown on liquid, but in our case, highly viscous substrates, create their own flows. And these have no, actually intrinsic motility. It's no flagella, like the kinds of things that Roman Stocker likes to study. And we did it actually with yeast, not bacteria. So here's what happens if you repeat that experiment I showed you on a hard agar plate with yeast, Pseudomonas, Saccharomyces cerevisiae, where you have two different heritable markers, yellow and red. The yeast expand radially. They segregate out due to genetic drift into these different sectors. There's this early stage of inflation where they're fighting it out, and eventually they decide locally, oh, I'm going to be red or I'll be yellow and so forth. So if I redo this experiment on an extremely viscous, but still finite, not infinite viscosity fluid, it's actually a standard medium, yeast peptone dextrose medium with some cellulose, so some branch polymer, various concentrations that allows you to tune the viscosity. And what's shown here actually, I think, is the viscosity for 600 Pascal seconds, the viscosity which equals 600 Pascal seconds. That's 600,000 times the viscosity of water. And already something has happened, even though the viscosity is only, slightly less, so to speak, than the hard agar. This early epoch of genetic demixing, when the two strains are fighting it out, has been stretched out in space and time. So that's something we'd like to understand. And to telegraph the main conclusion of these very nice studies by Severine and Brian, what we found was that without any motility, without any flagella, these yeasts, which have never had any motility as far as we know, anytime in their 2 billion plus years of evolution, they nevertheless generate a submerged vortex ring underneath the colony that stretches out the colony in interesting ways. And the way that was determined experimentally was to put in fluorescent beads. These are not reproducing organisms in the bulk of the of this very viscous soup on which the colony floats, and watching the beads move around. And that's how Severine and Brian discovered the submerged vortex ring. And so we don't need pumps and syringes. The colony itself generates flows that dilate the growing cell mass radially in ways that we will be discussing. So here's the early stages of what's going on in these experiments. It turns out that microorganisms, colonies floating on a viscous liquid, depending on the time scale and the conditions, can behave either like gases, liquids, or solids, except they're reproducing. So in my view, that makes them more interesting than conventional materials, conventional phases of matter. And what you're seeing here is them clumping up on a roughly 20-minute time scale before they ever have a chance to divide. These yeast divide every 90 minutes or so. And we believe they clump up because they are subject to capillary forces on the surface of the liquid on which they're growing and floating. And these capillary forces, as you can find out from my colleague Lakshmi Narayanan Mahadevan, is also responsible for the clumping of Cheerios, both in clumps and also attracting it gets attracted to the wall of this cereal bowl. And so there's something like a coarsening or spinodal decomposition that happens in the very early stages. But I'd like to talk about what happens later when things really start to reproduce and do interesting things. So here's a movie. This is happening over 14 to 20 hours roughly time scale. This is a movie of one of these inoculants of yeast floating on the surface of a liquid, as I said. And I want you to take a look at this. So there's this interesting texture. Eventually, out here, the organisms decide that they're either going to be blue or yellow. This is maybe 14 hours, I think, over which we've been watching this colony grow out in a radial circular fashion. But look at this texture and look inside this red circle. To my eye, that looks like a lizard. It's not actually a lizard, it's just a pattern of yellow yeast. But if I go backwards in time, watch the lizard, and you can see it shrinking, but preserving its shape. It's as if the lizard were painted on the surface of now a shrinking balloon, but now an expanding balloon. So the colony in its interior undergoes a conformal transformation, angle-preserving transformation that preserves shape. And so that's one thing we would like to understand. Here's our first cut at trying to understand this, this type of problem. We have our Petri dish, and we imagine that there's a liquid-like colony up here. It turns out to be about 200 micrometers thick. And then we look at what's the dynamics of this liquid-like colony, kind of quasi-2D liquid, floating on a liquid, another liquid, which is our medium with the cellulose in it. And now let's try to see what the fluid mechanics looks like for this colony. It involves both changes in the space and time dependence of the density. This is just the equation of continuity. And if there were no cell divisions, this would be equal to 0, standard fluid equation. But in fact, these are alive. And so what I put over here is reproduction rate for the cells in the colony. They could be either yellow or blue, for example. And it's not hard to show that with this kind of equation, there's a solution which has a radially symmetric angle-preserving velocity field, a dilational velocity field. Its divergence is equal to this growth rate, it turns out. And then the corresponding velocity field points radially outward and gets faster and faster as you go out from the center of the colony. And that gives us some understanding of how these conformal transformations of lizard-like shapes take place when we go forward in questions. Okay. Let me, any questions at this point? This isn't a bad place to stop.
I actually have a question.
Mm-hmm.
It seems like you have this very big surface with many different structures. I mean, is there a way to see if this would just be something that would happen statistically? I mean, it doesn't seem that unlikely that some of them are going to keep shape.
Well, okay, sure. In fact, if we look carefully, all of them keep the shape. And so it's a conformal transformation of the whole thing. And it wasn't just that we discovered, as you might worry, indeed, properly, that this was a lucky lizard that we found. They all do that until you get out to the edge. And then the system decides locally, I'm going to be all yellow or I'll be all blue, and then you start to get the beginning of sector.
Okay, that's interesting. It's really hard to see that on my screen, so thank you.
Yeah, I agreed. And of course, you know, I have a biased eye because I'm in love with this experiment, but this shape over here, if you can see it, is roughly the same, at least, as this shape here. And
so since I'm asking this, did you have to set up conditions very carefully for this, or does this happen no matter what when you mix the two together?
we can do, the nice thing about this is it's not like looking at plankton in the ocean where you're more like an astronomer and you're just looking
Right. Yeah, that's what I'm asking.
what you see. So we do it over and over again and we always see it.
Okay, thank you.
For sure. Any other questions?
David, I have a question about something you mentioned earlier on the spinodal-like decomposition.
Yes, Suraj, right.
Is there a version where you might see binodals and nucleation-like phenomena instead of?
Yeah. Okay. So the question was, instead of having just coarsening in the early stages before the cells start to divide, could there be nucleation phenomena? And I would say that there it probably helps to have antagonistic organisms. So you might know that Andrea Giametto is studying what happens when we have killer yeast that are creating poisons that cross-poison each other. And that produces the analog of a line tension. And then you can have that nucleation phenomenon. But what I called spinodal decomposition, it was a kind of just a loose term for the outcome of sprinkling them down. These are as genetically neutral as we can imagine. And so in that sense, there's no tendency to phase separate.
I see. Okay, thank you.
When they divide, on the other hand, out at the edge, they do something like phase separation, but that's not due to the antagonisms, that's due to the fact that they're dividing and there's genetic drift. Okay, so, yeah, go ahead.
Just to clarify, the yeasts have already been selected, which means the genetic competition already happens?
So yes, so we can, if you want, we haven't done a lot of experiments in this context. You grow these things up, they have a heritable yellow and blue strain, and we chose to inoculate them at a 50-50 concentration. However, we've also done experiments where we have 99% blue and 1% yellow, and then you can think of the yellow as being like a mutation, which is just occurs spontaneously at some point in the colony, and watch what happens there. So they've been selected to be as neutral as possible. They've been genetically engineered, thanks to the wizardry of Andrew Murray and the people in his lab, to have these heritable colors. So yellow begets yellow, blue begets blue, and it gives us a kind of, they're like tracer particles, they're like genetic tracer particles, so we can actually track the history of who begat whom, and you'll see that that's going to be important later.
I see. Thank you.
Great. Okay, so here's evidence there's actually something more. I think it was Lisa Randall who was worried about this, and indeed there may be something more, because there, in addition to the sort of outward pushing that produces that those kind of conformal transformation of the colony, there's this massive underwater, so to speak, vortex ring. And if you look at it in cross-section, if you measure it with this particle imaging velocimetry trick, it's radially outward, okay? And it extends far beyond the colony, far beyond what you would expect for mere pushing. Just the cells dividing and pushing each other out of their way, which is what that simple theory was suggesting. Here's a azimuthal average of what happens. This is the edge of the colony right here. And then things fall off. They fall off rather slowly. If there's a submerged vortex ring that is contributing to this flow, we can actually understand it pretty easily by just transposing the physics of anti-Helmholtz coils. So here's a coil with, imagine there's a current flowing. In actual fact, we're interested in vorticity, but the equations are very similar. And then we have a boundary condition at the air-liquid interface for our colony. In fact, even if the colony wasn't there, if there was a submerged vortex ring, you want the velocity field to be tangent. You don't want velocity to be going from liquid to air and carrying fluid out into the third dimension here. The equations, if I know that there's a vorticity concentrated on this ring, are that the curl of the velocity field is the vorticity, and it's a 3D incompressible flow down here. And we can just use the Biot-Savart law to find out what's happening, because these equations are nearly identical to Ampere's law and no magnetic monopoles for a B field. And this is an arrangement which is rarely used, I think, in experiment, but it's called anti-Helmholtz coils. And you can show that there's a fall-off, a power law fall-off of the velocity field, which seems to match roughly, at least, with what's going on in the experiments. So there's something else going on. We haven't explained it yet, but there's a submerged vortex ring that seems to be giving rise to interesting effects. So in fact, you can work out what happens, and there's another radial contribution, another dilational contribution to what goes on in the stretching of the colony just due to this submerged vortex ring. So we thought long and hard about what's going on, and we had distinguished fluid mechanicians, some of them at Harvard, saying, you guys are getting flow, you do your best in the lab, but maybe there's some stray thermal gradients, and you have Rayleigh-Bernard convection. Well, Brian and Severine redid the experiment without the colony. They're very careful. Nothing happened, no stray thermal gradients. Then there's something called the Marangoni effect, which has to do with surface active molecules that might be secreted by the yeast. And then they would maybe be pushing the colony apart and contributing to the fluid mechanics at the surface. And Severine and Brian did something very clever. They did a lot of tests, many tests. They grew the colony not only at the top surface, they grew it on the wall, they grew it on the bottom. They still got a submerged vortex ring down here at the bottom when there is no surface active molecule we know of that's working. And they get an interesting vortex ring that actually tends to compress the colony. And then they did an experiment, which I like a lot, that sort of reveals a bit about what's going on. And let me see if I can get out of this in some easy way. I want to be able to get rid of my laser pointer and start this cool movie. So what are you seeing? You're seeing a colony at the bottom. You're seeing these little green beads that are just there as themselves as tracer particles, non-reproducing tracer particles, to tell you what's going. You see them moving around. So let's see what happens if I run this thing forward. You can sort of see just this little slow motion of these things. But then starts belching out gas bubbles. Now those gas bubbles, we believe, are carbon dioxide. And in fact, there's metabolism going on down here at the bottom, largely anaerobic, which is what yeast do best. They make ethanol. They're used to make beer, after all. They convert dextrose via this interesting metabolic pathway into ethanol plus CO2. So CO2 is coming off, and I'm not going to give you a theory of what happens when the colony is on the bottom. I'm going to care about what happens on the top. But this is an experiment that's kind of clue that tells us that metabolism is important. Okay. So in fact, we can redo the experiment at the top. There's an aerobic metabolic pathway that also produces ethanol. Very clever yeast. And if there's any CO2, here it is, it'll go off into the atmosphere, not cause this major belching that we just saw. And now we said, well, maybe we better think about the metabolism and see how it might change the fluid dynamics. And the basic equation, actually, I just derived this equation this morning in Physics 268 BR. It's an equation to which I will be applying the renormalization group later this week. But for now, let's just think of it as an equation that describes what goes on in this very viscous liquid. So just F equals ma, but written out in this non-trivial way with this nonlinear term here and so forth. And then we also tracked the density of the medium. And what Severine and Brian discovered was that the colony takes up the dextrose, and the left behind liquid after the colony metabolizes it and uses it to grow, etc., is lighter, about 2% lighter, let's say, or 1.5% lighter than it would be normally. And in fact, as a result, you have to take into account buoyancy effects. The colony actually ends up being lighter at the top and heavier at the bottom. At first sight, that doesn't look like that helps you because that's a stable situation if it's heavy at the bottom and light at the top. But if you put in this little term here that allows you to model the uptake of the dextrose, here are some boundary conditions. You can actually make a weather map of what's happening under the colony. So here's the colony, here's the liquid in the Petri dish. These green bars are the lines of constant pressure. And the pressure is going up like any self-respecting fluid under gravity as you go downwards. And then these red lines are the lines of constant density. And it's lighter here, and it gets denser and denser as you go into the deeper into the fluid where you have the unharvested dextrose, and so it gets lighter up here and it's heavier here. So with this weather map, we can decide whether this fluid is going to want to flow. And the easy way to do that is to take the curl of the Navier-Stokes equations. And you'll get something that looks like this, with the vorticity. This is the measure of the swirliness of the liquid. And this is just an advective derivative of this vorticity field, which may or may not be coming up here underneath the colony. Here's the edge of the colony. And if you do that under conditions where there can be a small change in density, these nonlinear terms were at very low Reynolds numbers, so they can be neglected. But the important term for us is over here on the right. You get a term which, where you take, you end up with the cross product of the gradient of the density and the gradient of the pressure. If there's an equation of state, this would never happen. These would be parallel and this term would be absent. But sure enough, in Landau, Lifshitz, and other books on fluid mechanics, you can find this effect. And whenever you have a cross product of the density gradient, which you can see is pointing down like this, and a pressure gradient, which you can see is pointing straight downwards, you'll have a vorticity mill, a vorticity source term in this equation. So this initial condition is unstable to the generation of vorticity. I invite you to work out, using the right-hand rule, the sign of the vorticity, and it goes around and around like this. And there's azimuthal symmetry, so I've described the creation of a vortex ring. There's some diffusion of vorticity as well, but as Brian showed, if you do a simulation under these conditions, you end up with a submerged vortex ring that is very close to what's observed in the experiments. And this is actually a thresholdless, it's called the baroclinic instability. It is responsible for a lot of the weather. And if you turn this on its side and look for these crossed lines, isoclines and isobars, you can understand how hurricanes, a little bit how hurricanes are generated, maybe possibly tornadoes. In any case, we think we understand where this ring of vorticity comes from. I don't have time to do this. This is a nice simulation by Vamsi Spandon, advised by Michael Brenner. I'll just show it maybe quickly and see if I can do this easily. But this is a growing colony, and you can see what happens to both the vorticity and the actual density. And sure enough, you get, with the growing colony, a submerged vortex ring whose center of vorticity tracks the growing colony. Okay, so I've done the simple part of this problem in the sense that we had mostly a viscosity which is 600 pascal seconds. That's very, very viscous. I now want to start asking what happens if we take all the other conditions more or less the same and we simply reduce the viscosity by knocking down the concentration of cellulose in these experiments. And so over here, this is very much like the experiment that produced the lizard, but you'll see that something remarkable happens at the end if we reduce the viscosity to say 450. Some of the yeast decides to leave the mothership. Still stays at the surface, but it starts to migrate out to the Petri dish, and it does this without any intrinsic motility, no flagella or cilia or anything comparable. And if you knock down the viscosity a little bit further, you'll get this remarkable movie, at least I think it's remarkable, on the right, which to me looks like the 4th of July. And in fact, this instability of the yeast, this growing yeast, obviously has a pretty profound effect on the population genetics. It sort of helps the system segregate out into blue genotypes, yellow genotypes, and pink genotypes. And it also allows it to colonize the entire Petri dish, I don't know, maybe roughly in 12 hours instead of the normal 3 days or 5 days it would take. So it's a way of colonizing, exploiting the submerged vortex ring, we believe. In fact, the submerged vortex ring, in some sense, is ripping the colony apart. It's a liquid colony, so you get these long tendrils coming out. And there's a beautiful black and white movie that I hope to show here, which shows one of these tendrils. So this tendril up here, let's say we transpose it down to here. It's pointing out from right to left. And it's going to be growing out, ripped away from the mother colony by that submerged vortex ring. And well, you can see, look, there it is, pinching off, pinching off, pinching off. It looks like a little bit like the instability that gives rise to the droplets on a spider web, droplets of dew in the morning, perhaps. But unlike dew droplets, this material is alive. These are actively dividing cells. And so when it tries to pinch off, the cells are desperately reproducing, trying to resist this pinch off. So there's clearly issues involving surface tension, but also we need to combine traditional fluid mechanics with life, with the ability of these cells to divide, and of course, to understand, hopefully, this genetic demixing. If I'm going to show this fireworks display, could be Bastille Day, in fact, one more time. And here's some simple theoretical ideas. Okay, most of this is experiment, and I'm certainly not at all a card-carrying experimentalist. I only do experiments that are so easy that even I can do them. But here's some theory that we came up with that accounts for some, might account for these liquid fingers, this fingering instability. So here's our colony. And what we decided to do theoretically was to track the height of the colony. And of course, as the colony gets stretched out, it's going to thin. It'll desperately maybe try to maintain some equilibrium thickness, but by cell division. But as it gets stretched out by the flows that it induces, the submerged vortex ring, there's a competition between the growth of the colony and this thinning effect. And so you can understand thinning by just saying, well, the height of the colony is a dynamical variable that gets advected along by whatever velocity field is inside the colony. We'll take this velocity field to be the sum of those two effects, the colony is just pushing things out and the effect of our metabolically generated vortex ring. So we'll put some coefficient alpha here. Alpha will get stronger and stronger as the vortex ring gets more and more violent. How do we make it violent? We make it violent by turning down the viscosity of the fluid and thus getting a more vigorous vortex ring swirling around here. This diffusion just basically says the colony tries to be flat locally. You can think of it as like a line tension if you prefer. And here's a kind of logistic equation where the height grows up because the things are reproducing, but there's some equilibrium height, H naught, because the nutrients don't allow you to get colonies. You can't get nutrients up into the colony that's thicker than H naught. So this is a nonlinear partial differential equation. It looks a little bit like something called a Fisher equation, but it has advection. And in particular, it has this dilational velocity field, the same thing that produced the conformal transformation on that lizard. And you can show that there are wave-like solutions, even though it's a parabolic partial differential equation. There are wave-like solutions nonetheless. And you can track the height and you contract the radius of this thing, and you find that the radius under these conditions is actually growing exponentially in time, unlike a conventional Fisher wave. All the cells are actively dividing here under these conditions, and you end up with exponential growth, which is sort of what you might have expected. But the interesting dynamics is in this height. And if you look carefully, this height variable's fate depends crucially on the cell division rate of the organism, in this case Saccharomyces cerevisiae, yeast, and the thinning effect of this alpha term in the dilational velocity. And if you solve this equation, you find two regimes. One is where the thinning can't keep up with the growth, and you end up with a height that's reduced relative to what you would have in the absence of metabolism-induced flow, or you can actually drive the height to zero. We call that the thinning catastrophe. That's illustrated here. Brian solved the relevant differential equations. If there's no growth, we just have height coming out. This is a standard Fisher wave. These are equal time intervals, and it goes at a constant velocity. But as soon as I allow some actual thinning, where we have a non-zero alpha, the alpha is 1 per hour, here it's 2 per hour, the height goes down. That's one thing that happens. It starts to thin a bit. And then it actually starts to grow exponentially, in fact. And then, both here and here, I get a height that drops down, but there's a critical value of alpha beyond which the population just collapses. Now, these are not atoms, or of course, that are spreading out and reproducing. There are cells. They are cells that are about 5 microns in diameter or radius. And these 5 micron reproducing objects define a minimum thickness for this, this colony. And so we think that when you get near this thinning catastrophe, the system has no choice but to break up into these long little filaments because now I get a reduced average colony concentration, and I don't have to drop below a colony that's 1 cell thick. You can't go thinner than that. We're not sure that this is the explanation. We're hoping to get help from people like Vamsi Spandan and Michael Brenner. Howard Stone at Princeton is also interested. But that's what we think is going on. I want to conclude by just showing you one more thing, which is what happens if we take the viscosity way down to 85 Pascal seconds, maybe 8 times smaller than what I started with. So now it's a very violent situation for the, a very violent circumstance here. Let's see if I can get my laser pointer turned off. Very, meaning 8 times as violent as what I started with. And here's a colony, and I want you to watch what happens when we have a particularly vigorous submerged vortex ring. Now the colony literally rips itself apart. That's so cool, I want to show it again. In fact, this is a little bit like plate tectonics. Here's South America, and here's Africa. And now the colony is behaving in a viscoelastic way. The flows are so rapid, not literally rapid. The eddy turnover time might be 6 hours or something under these conditions, but rapid enough so that the colony growth cannot keep up with it, and it behaves, instead of like a liquid, it behaves like a solid on an experimental time scale. And there's another white light, very beautiful movie, which shows in detail the breakup of these continent-like structures when you have a particularly violent submerged vortex ring. Here it is breaking up. And you see these tendrils. Those are not the typical material science tendrils, even though there was some material science support for these experiments. Those are alive. and they are attempting to bridge these gaps by having cell division. They're unsuccessful in this case, but it's a whole new twist on fracture dynamics, where it's the fracture dynamics of something living. So here's one other, almost the last slide here. If it's true that one isolated colony has a submerged vortex ring, all these fragments should have a submerged vortex ring under them. And you can see that's consistent with the accumulation of these yeast fragments on the midplanes between these continents. And we've now circled back to these accumulation points that are associated with the downwellings of microorganisms in the ocean. These are accumulation lines, of course, for yeast. So with that, here's a summary of our results, some of the things I've told you about. These are the people who did the work. So I'd like to pause now and thank you for your attention. Happy to take more questions. I'm happy to hang around for a while if there are people who would like to ask things. Thanks a lot.
Thank you, David. So please raise your hand if you have questions. Benjamin Maisel.
Hi, yeah, I have two questions. The first is, in the metabolism, you considered how it was consuming nutrients, but can the metabolic reactions also be, for example, exothermic and release heat into the environment?
They can, and we worried a lot about whether there would be temperature gradients at the top of the colony that would maybe change things or create instabilities and so forth. As far as we can tell, by using various kinds of estimates, those temperature gradients are extremely small. Moreover, a lot of heat, when it's done at the top of the colony, is going to be released into the third dimension. So as far as we can tell, there are no measurable temperature changes due to exothermic reactions for this particular experiment, but there could be other circumstances where that's important.
I see. And my second question, in this final example of a very liquid, low viscous substrate on which they grow, is there some critical size of these patches at which point they start generating their own vortex ring and tearing themselves apart?
There probably is. We haven't studied that in any detail, but there probably is something like that going on when these colonies start to break up. When they start in the circular form, there's a single submerged vortex ring, and at some point they have to their metabolism will reorganize, and you'll get submerged vortex rings about each one of these continents. At least that's what we believe.
I see. Thank you.
Right.
Okay. Suraj Shankar.
Yes.
Hi, David, again. I had a question about the last, the picture, the video you showed about the solid-like fragmentation, which is incredibly beautiful. So most of what you told, if I understand correctly, had to do with an interplay of two timescales. One was the growth division-induced timescale, and the other came from the vortex ring and the flow generated.
Yes.
Is there an additional timescale that controls how liquid or solid-like the colony is?
Yes, there undoubtedly is. Thank you for the question. There certainly will be some sort of timescale like that. And there's a viscoelastic time, and you probably know that there's a Maxwell term that you can write down for viscoelastic solids. So that introduces another timescale into the problem, above and beyond the reproduction time and so forth. But it seems clear to me that all these wonderful instabilities that have been known about over the history of the Navier-Stokes equations and solid mechanics equations, such as the Rayleigh plateau instability, where you get droplets, they need to be looked at again with cell divisions. And there's probably a time scale, and Jim Rice would know a lot about this, our colleague in Cs, for a fracture and this Maxwell time. But we would want to add in the possibility of cell divisions and see how things change. And it suggests, I think, a number of fruitful areas for investigation.
Next we have Amir Vikram.
Hi, thank you for a very interesting talk. I realize this is going to be extremely speculative, this question, but do you have any thoughts whether yeast or any other organisms could have evolved somehow to optimally take advantage of these sorts of fluid flows that are created, be it in their cell morphology or otherwise?
That's an interesting question, and we've thought about it. And you can ask, will yeast perhaps somehow want to exploit this strategy to spread across sap coming out, a very viscous sappy resin coming out of a tree or something? The short answer is I don't know. It would require further investigation. I would like, even though the Reynolds number is very different, to know whether there on average, there's some submerged vortex ring underneath these plankton blooms in the North Sea or off the southern coast of Iceland. For similar baroclinic reasons, it's possible professional oceanographers already know about this. In any event, we have a laboratory example, and I don't know if somehow, how often yeast in its evolutionary history has exploited this. It certainly wouldn't happen if you didn't make the fluid on which you were floating less dense. And if you somehow had enough waste products that made it denser underneath you, then something different would happen. So, interesting question. I don't know the answer.
Zechen Zhang. Oh, okay. Then I have.
Oh, sorry.
Go ahead, Zechen.
Sorry, I forgot to unmute. Thanks, Professor Nelson, for the talk. I just have a question about the viscosity. So in your solution, so in your theoretical toy model, the solution depends on alpha that you imposed, but it doesn't explicitly depend on the viscosity. So I was a little confused.
Okay, so there was an implicit, thank you for the question, there's an implicit viscosity dependence because in particular, on both the viscosity and the depth of the fluid. So one of the things that I think Severine in particular was involved in was making the depth of the fluid shallower and shallower. And that tends to squeeze out the vortex and makes it more difficult to form. Conversely, you could make it higher, and then the vortex ring can more easily form, and you can also control that by the viscosity. So the idea is that in that alpha term, there are two contributions. One is just the growth rate of the cells, but the other, which comes from those that anti-Helmholtz coil model, has to do with the intensity of the flow at the surface of the colony that's ripping it, that's really playing probably the most important role in ripping it apart. And so that will be strongly viscosity dependent. So imagine the alpha has, in its argument, it will be alpha of viscosity, and the low viscosity will be larger alpha.
Right, so you are not interested in calculating alpha directly from the...
Well, we can estimate it, and that Helmholtz coil argument lets you get an estimate for the size of alpha. We have estimated it in our which you can read about if you want in PhysRev X, and it seems to be in rough agreement for our experimental parameters. But it certainly will be viscosity dependent, absolutely.
Thank you.
Subir, you're muted.
Sorry. Sorry. There are several more questions. But before I call on the questions, let's just all thank David for a great colloquium. And so you can actually or virtually scrap and leave if you have other obligations. And then we'll just go back to the questions now. So Minchol Park.
Yes, thank you for the great talk. And I just want to ask about if I understood correctly, then the cause of the flow is the gradation vector of the fluid density and the pressure. So how does the metabolism affect this gradation vector of the fluid density? So it was my question.
Yeah, okay. So the way you get a gradient underneath the colony, and the gradient lines of density bend around. They bend upwards as you get in the fluid, as you get to the edge of the colony. The reason there's a gradient is that these dividing cells are constantly sucking fairly heavy sugar molecules out of the liquid. And when they do that, the liquid that's left behind in the vicinity of the colony itself is a little bit lighter. And that's what produces these curved contours of constant density. They're sucking the dextrose out of the nutrient liquid on which they are floating, and they are, through the beautiful, complicated, intricate metabolic pathways of life, producing two cells where we had one before. Maybe they would use 50,000 dextrose molecules to do that. But in any event, the fluid underneath is a little bit lighter, and that's what drives this density gradient. The pressure gradient is the usual hydrostatic pressure gradient. And because they're at an angle, as I said, you get this a thresholdless aeroclinic instability. You start generating vorticity sort of automatically in the fluid. At least that's our theory.
Thank you.
Yep.
Then we have Maha.
Hi, David. This question just follows up on the answer to the last one. It's not obvious to me that it should be thresholdless, the paraclinic instability. Maybe I missed something because you said that there is a rate of uptake which essentially gives rise to this buoyancy.
Yep.
And then there is a effective momentum diffusivity, which is essentially trying to equilibrate viscosity. And
Yep.
so is there, again, a ratio of timescales that is appropriate so that you don't essentially have any threshold whatsoever? So as soon as you get grad P not parallel to grad rho, you immediately get something? And maybe a related question is,
okay,
metabolically, can you tune the rate of metabolism so that you can start to see when you will actually get a threshold?
Well, I'd have to ask our colleague Andrew Murray whether there's some ingenious way to tune the metabolism. I mean, you could starve it, of course. You could maybe cut down the dextrose concentration. We always tried to have an abundant amount of dextrose. I mean, thresholdless may be a misnomer, but there are people who call it that, I think. But you're right, vorticity can be diffused away from where it's created. But there's always a source term on the right-hand side of that vorticity equation that I had before when the lines are not parallel. And, I suspect, you would know better, Maha, but, if you look at the birth of a small tornado in the atmosphere, you would probably have similar kinds of questions. But what I can assure you is that with the parameters that Brian extracted from his experiments with Severine and then simulated, that we're well above any threshold. If there is a threshold, it's pretty small.
Thank you.
Right.
Okay, I actually have to leave now, David. So yeah,
no problem.
The names there, Core Park, probably the next one. And so thank you again.
Sure.
Park, and go ahead.
Okay, thank you very much for your talk. I have a very simple question from the last slide. I see that the colony starts growing, but it can't overcome its own growth and just splits into many.
Yes.
So what happens after the video? Does it reach a steady state, or does in special conditions, can it go into a dynamically, like, keep changing state?
Okay, so if we had set up, thank you for the question. If we had set up the equivalent of a chemostat, where we were constantly providing nutrients, then I think what would happen is the colony would just thicken and become some kind of yeasty sludge on the top of the liquid.
Okay.
We don't, in fact, do that. We have a finite amount of nutrients. And I think for the runs that Vamsi Spandon did with approximating our experimental conditions, eventually the colonies, the fragments, or the colony, if it doesn't fragment, runs out of nutrients and just stops growing.
Oh, okay, that makes sense.
So that's what happens under our conditions. Anyway.
So it might happen if you keep feeding nutrients.
Yes, you'd have to maybe have some plumbing.
Okay.
Sharad Ramanathan has occasionally been interested in having plumbing in these Petri dishes where you constantly supply nutrients.
Okay, thank you.
Right, and I see a hand up from my colleague, Ike Silvera.
Hi, David.
Hi.
I was wondering, did you try varying gravity? Because that would change your pressure and it would change some of your parameters, and you might find it on different planets.
Well, if perhaps you can, we can team up to submit a proposal to NASA if we ever get back to normal, and we can do it on the space shuttle or something. But there are other. We could also change there are other ways, less expensive, of changing the density of our fluid. In fact, the yeast is slightly heavier than I believe, than the liquid on which it sits. It's just a line tension, or surface tension rather, that keeps it from breaking through. Eventually, that's another thing that might happen in the later stages. So yeah, we could try that. That would be an interesting parameter to change that gradient of the pressure, but the gradient of the density is probably easier thing to go after. And then, as Maha suggested, change the metabolism, because that certainly would have an effect. And we can just do a very crude thing, as I think I mentioned briefly. Brian and Severine just let the colony eat up everything and weighed the resulting liquid, and it was say 1.5% lighter. And if we could find a way to vary that, we'd have another way to test the theory.
Okay.
All right, and I see a question from Amala. How are you, Amala?
Hi, David. Thank you very much for that talk. So the baroclinic vorticity generation term is interesting. I just wanted to make a comment that baroclinic instability is actually a term that's used for a different kind of instability. It involves the rotation of the Earth, and the vorticity that you end up with is in fact normal, or in the perpendicular to the vorticity that you're showing. So it's more a question of semantics, that baroclinic instability gives rise to some of the weather systems and storms that we see, but that is in fact very different, and the direction of the vorticity is completely different than the vortices you're describing. So.
I agree, and you would certainly know better than I, but I believe I extracted the term from, I may have extracted it from the Landau-Lifshitz book on fluid mechanics, where it does have the form that I...
Yes, it's the baroclinic generation of vorticity is correct, but it's not the baroclinic instability.
Oh, okay. I will be cautious in the future. Thank you very much for that.
Thank you.
Okay.
David?
Yes, please.
Just a question. Is it simply the alcohol that's being released by the yeast that's reducing the density of the
It's a combination of things, and I'd have to go back and check our paper. It's two things, right? Absolutely, there are, you're taking up dextrose. You're doing a lot of stuff to make the colony more massive because there are more cells, et cetera. And then there are, you might call them waste products, but of course, if it's alcohol or ethanol, we might not consider them.
Some of us might not consider that waste product.
That's right, exactly, especially during these difficult times.
Right.
So the net change in mass is a negative sign. So whatever it takes out, it doesn't give back, it gives back less in mass than what it takes up. And so, well, maybe some of the alcohol evaporates into the third dimension. The CO2 probably does. So we haven't tracked down everything, but we do know it's lighter, and it's probably because that missing dextrose is not completely compensated by whatever is extruded like ethanol from the colony. So you're muted, Bert, if you're replying to me. But...
No, I wasn't saying anything.
Okay. All right. I don't see any more hands up. So if I don't see any hands, unless Amala put up her hand earlier, I will thank you all for coming, and thank you all for your interesting questions. I hope I've maybe indicated that there's stuff in simple biology that is tabletop in nature, and even the early experiments could be done by an incompetent theorist like me. So maybe there's a few low-hanging fruits here that might be of some interest. Okay, so I'm going to leave. If anybody has any further questions, don't hesitate to email me, and look forward to interacting with all of you in a more normal way in the future. So, see you.
Thank you.
Right, you're welcome.
Bye, David.